As we continue to move through Chapter 2, entitled Water, Biochemistry, and Cells, I'm now going to highlight the section covering the structure and function of macromolecules. Macromolecules can be divided into four main groups. We have the carbohydrates, the lipids, the nucleic acids, and the proteins. For each of these, we're going to talk about the monomeric form and the polymer form. So a polymer is basically just a repeating unit linked by covalent bonds. So here depicted as a string of A's. The monomer is the individual repeating unit that makes up that polymer, so the single A. So as we begin to study macromolecules, you need to have a working knowledge of both the monomeric and the polymeric form for the particular macromolecule that we're discussing. So for the case of carbohydrates, we know that these are basically just a string of sugars. So the monomeric form would be a monosaccharide, mono being one, a single sugar, the function of which is for energy storage as well as structure. Nucleic acids, which are DNA and RNA, are made up of nucleotides, and the function of which is to store information. Proteins are comprised of one of 20 amino acids. The function of proteins are for various enzymatic reactions, and they also provide structure. Lipids are a little bit more complicated. They're a very diverse group of macromolecules. They're made up of things such as fatty acids and glycerol backbones, and then their polymeric form can include things such as fats, lipid membranes, various steroid hormones as well. So it doesn't fit as nicely into our table. So let's start with a discussion of carbohydrates. The structure of carbohydrates is comprised of carbon, hydrogen, and oxygen in a very specific ratio. And carbohydrates are everything from table sugar to potatoes, your grains, and breads. So let's begin with the monomer of a carbohydrate, which is just a monosaccharide. And here you can see this is your simple sugars and would be defined as something as simple as just a glucose molecule. If we were to combine two monosaccharides together, this would be called a disaccharide, di meaning two. Here is demonstrated a sucrose molecule, which is simply the combining of a glucose and a fructose molecule together. Polysaccharides, poly meaning many, are many monosaccharides linked together, and here you can see cellulose is a string of glucose molecules bonded together covalently. So what are the functions of carbohydrates? They are our main energy source. They also are involved in energy storage. So if you take in more energy than you need, the excess energy will be stored in your cells. They also, for plants, are involved in forming their cellular structure and things such as cellulose. So if you had a hamburger and fries for lunch, you just ingested a large number of carbohydrates, which ultimately is made up of glucose. So upon consuming that food, what you will find is that your blood glucose levels are going to go up, as shown in the middle of this diagram. What that ultimately does is it activates your pancreas, which then will release insulin into your bloodstream. The insulin will then signal to all your cells that glucose is available, and glucose will then be taken up into the cells throughout your body and driving different energy needs appropriately. If you take in more glucose than the cells in your body actually need, what happens is that excess glucose will then go to the liver and get stored in the form of glycogen. Sometime later in the afternoon, when it's been a couple hours since you last ate, what's going to happen is your glucose levels are going to start to drop, and you can then initiate the release of glucose into the blood from the glycogen that had been stored in your liver. This will then cause your blood glucose levels to normalize once again. So that is a brief overview of carbohydrates, which, as I mentioned, is necessary for energy use, and it's also important for storage and cell structure. Now let's discuss proteins. What are proteins? Well, they're just basically a string of amino acids that are joined together by a peptide bond. They are made up of carbon, oxygen, hydrogen, and nitrogen. You'll recall from the carbohydrates that they did not include nitrogen. There are 20 different amino acids, each of which has a different chemical property. So what are the functions of proteins? There's a vast array of functions of proteins in your body. They can be involved in everything such as structural proteins, all the enzymes that drive the various chemical reactions that you need within a cell. They're involved in antibody production, the formation of blood clotting factors. They're your cell receptors that are waiting at the plasma membrane. They're involved in the formation of transcription factors, which is what drives the expression of genes and turns genes off and on. So proteins is one of our most diverse macromolecules that we have. Here is a slide showing all of the biochemical pathways in our body, and there is a different enzyme and hence a different protein driving each of these reactions. So each of the words here shown in blue is a different enzyme. I've included this slide just to show you that we have a diverse array of enzymes, and each of these are specific for that one particular chemical reaction. So that is a very brief overview of proteins. Let's move on to lipids. What are lipids? Well, they are hydrophobic, meaning they fear water. They're composed mostly of just carbon and hydrogen. Here you'll notice that it does not include oxygen. There's three main types: fat, steroids, and phospholipids. Fats are composed of a glycerol molecule joined with 3 fatty acid tails. Steroids are basically 4 carbon ring structures, things such as cholesterol, estrogen, testosterone, glucocorticoids, etc. Phospholipids are composed of glycerol molecules, 2 fatty acid tails, and a phosphate group, and I'll show you what each of them. So here we have fat up at the top, which, as I mentioned, has a glycerol backbone and 3 fatty acid tails. So you'll oftentimes hear these referred to as your triglycerides. Cholesterol is shown on the right with the steroidal rings. And then lastly, we have phospholipids, which also have the glycerol backbone, 2 fatty acid tails, but it also has a phosphate head group. That phosphate head group actually is not hydrophobic, but rather hydrophilic, meaning it loves water. So the phospholipid is an interesting molecule because it has half of a region which loves water, and the other portion of the molecule fears water. So what are the functions of lipids? Mostly they're used for energy storage, cushioning and support, as well as insulation. We have 2 major classes of fatty acids. When we're talking about fatty acids, we have those that are saturated, and when we say saturated, it just means saturated fully with hydrogen. And we have unsaturated fatty acids, which indicates that there is basically a double bond present, and it creates a kink in the structure of that fatty acid. Our saturated fats, as shown here, are all single bonds, and they pack very tightly together. They tend to be solid at room temperature and include things like butter and your solid fats. All animal fats tend to be saturated. Our unsaturated fats, as I mentioned on the previous slide, have double bonds in the fatty acid chain, and so they have these kinks in their chain, and this makes them less able to pack tightly together. So these are going to be your liquids at room temperature, things like vegetable oils, olive oil. Plants tend to be unsaturated. So that is a very brief overview of lipids. As I said, it includes carbon and hydrogen, and it's necessary for energy storage, cushioning, support, and insulation. So let's end our discussion of macromolecules by briefly covering nucleic acids. What are nucleic acids? Well, they're just make up polymers of nucleotides. What's a nucleotide? Well, a nucleotide is made up of a sugar, a phosphate, and one of 4 different nitrogenous bases. The sugar can either be a ribose or a deoxyribose. If it's a deoxyribose, it will make up DNA. If it's a ribose, then that will make RNA. So here you can see the ribonucleotide, which is made up of ribose sugar, is connected to a phosphate and the nitrogenous base. On the bottom, you can see a deoxyribonucleotide, which is basically a deoxyribose sugar connected to a phosphate and a nitrogenous base. So the ribonucleotides are what we find in RNA, whereas the deoxyribonucleotides are what you'll see with DNA. So DNA, we know, is what ultimately is necessary for the storage of all of our genetic material. It's our hereditary material, and the structure of DNA is made up of a double helix. So that concludes our very brief introduction to macromolecules. We talked about carbohydrates, proteins, lipids, and nucleic acids. Thank you so much.